Okay, so at the base of the heart, we have the blood vessels, the great vessels that attach. And then veins attach into the atria to deliver blood into the heart, and arteries attach into the ventricles to deliver blood either into the lungs, the right side, or the left side into the body. Now, into the right atria, it receives blood from the systemic circulation, which is deoxygenated, from the superior vena cava, inferior vena cava, and the coronary sinus. Left side receives oxygenated blood from the lungs, from the two pulmonary veins from the left side and the two pulmonary veins from the right side. Occasionally, we can have anatomical variations in which instead of having two pulmonary veins on the right, the person has three. Does that person has more oxygenated blood than others? Who knows? Because he has 5 pulmonary veins. Anyway, on the left side then, in the left ventricle, we have the attachment of the aorta, the main artery in the body that delivers the oxygenated blood into the body through its different branches, including the coronary arteries, left and right, that provide blood supply to the heart. And then on the right side, we have the pulmonary trunk that delivers the blood, the oxygenated blood, into the lungs via the right and the left pulmonary arteries, which are branches of the pulmonary trunk, so that the blood can be deoxygenated. And there is anatomical differences between the atria and the ventricles. Some of it will be the size. The atria are smaller than the ventricles. And the atria are located superiorly, so they are above the ventricles. So in terms of size, atria are smaller than ventricles. In terms of walls, the walls of the ventricles are thicker than the walls of the atria. The atria pretty much are passive chambers. They receive the blood supply, and yes, they can pump it to the ventricles, but since the ventricles are located down, even by gravity, just by opening the valves that we have between the atria and the ventricles, the blood can go down without so much effort. But the ventricles, they have to pump the blood to bring it up so it can exit through these great blood vessels. So the walls, they have to be more muscular, much thicker. And the lining of the heart, which is the endocardium, it has basically a smoother surface in the atria than the ventricles. So there are some extensions of muscle within the atria that are called pectinate muscles. And then in the ventricles, it's called trabecula carni. So the inside of the ventricles is rougher, and the inside of the atria is smoother. And specifically in the right atrium, you have more pectinate muscles than in the left. The left is smoother between the right and the left side. And then the atria has muscular pouches that are called auricles. Auricle stands for ear. Auricular region, the ear region. So they have like little ears, which are the auricles. So they can expand to provide more space for receiving more blood in some cases, not always. And then the septum that separates the right and the left atria is called interatrial septum. Before we're born, it has an opening that is called the fossa ovalis. And then when we close, we call it foramen ovale. Once we're born, we take our first breath, and then it closes. Then there is no shunt between the right and the left side of the heart, blood. Why is this? Well, because before we're born, our lungs are filled with a fluid that's called amniotic fluid, so we don't have oxygen available. So there is no purpose for us to shunt the blood only to the lungs because it's not going to oxygenate there. Who oxygenates the blood of the fetus? The mother. Mm-hmm. So then, that's why we have this, to shunt the blood between the right and left side. Now, the separation between the right and left ventricle internally is called the interventricular septum. And it starts forming from the apex up to the base, in between the atria and the ventricles. So, as it's forming and going up to close the right and the left compartments of the ventricles, sometimes it can stop before reaching its full edge. And the person is going to have something that's called ventricular septal defect, or VSD. Depending on how large is that defect, that opening, is whether you have a loud murmur or an extra sound of the heart, or a soft or less loud murmur. So as the blood, in the case of someone who has an opening in between the ventricles, as the blood passes from right to left, it will friction against that opening. So if the opening is small, how large is going to be the friction of the blood against that opening? Large or small friction? Large friction. It's like if this space, guys, was very narrow and you try to push an object against that narrowness of the space, you will hear a lot of sound. So the murmur is going to be loud. But if the gap is large, there is not so much resistance or not so much friction. So then in that case, the person won't have a murmur. But since there is going to be more deoxygenated blood from the right side to go to the left, the person is going to look a lot bluish. And in that case, it requires an urgent surgery. If the opening in the ventricle, interventricular septum, the VSD is small, sometimes the person can live without a surgery, and it will have just the murmur, because there is not going to be so much dilution effect. Not so much deoxygenated blood goes from the right side to the left. Does that make sense? So once a baby borns and you see a lot of blue color in their skin, that means something is going wrong with the heart, more than likely. Okay? So, this is to show you a section from the heart cross-section, as well showing you some differences between the ventricles. So this is the interventricular septum. This is the left side of the heart. So this is the left wall of the left ventricle. And then this is the right wall of the right ventricle. So see how thin it is, this wall from the right ventricle in comparison to the left. Why do you think is that? Why the right ventricle has a thinner wall than the left ventricle? Yeah, it's related to pumping. So which one has to work harder? The left or the right? The left side. Why? It has to pump the blood up to where? Up to your toes. So it takes longer distance. So it has to contract stronger, the left side. The right only has to pump the blood up to where? Up to the lungs. So it takes less distance. So it has to work less harder. So that's why this wall is thinner than this. So besides the left ventricle having trabecula carni, it also has some muscles that are called papillary muscles. These papillary muscles are finger-like projections that attach tendons, collagenous tendons that we call the chordae tendinae, the strings of the heart. These chordae tendinae holds leaflets or flaps of a valve, V-A-L-V-E, valve. So this valve acts as a one-way door that allows the blood going from atria to ventricles and not the other way around. So that prevents backward flow of blood. And then this chordae tendinae, which is attached to the edge of the papillary muscles and to the base of the cusp, it prevents the flipping of the flaps of the valve to stay in the atria. Because when the ventricles contract, they increase so much the pressure inside the chamber that the flaps tends to go up into the atria. And when these papillary muscles contract, they bring it down into the ventricles. So it's like, I compare this to an umbrella during a windy day. If someone is having an open umbrella during a windy day, the fabric of the umbrella will tend to go inside out. And then those wires that holds this fabric into the stem of the umbrella will be compared as to when you try to unfold this opening of the fabric back in. So you just pull it. And the papillary muscle will be basically your hand pulling those wires so they can go back to normal. Okay? So that's the function of these two these muscles and the chordae tendinae, these two structures. And then this interventricular septum separates both left and right ventricles so that you don't have shunt of blood. You don't mix the oxygenated blood with oxygenated blood. So here is the internal structure of right atrium, left atrium, smoother walls, smaller cavities. Then the ventricles, which are here down. This is the interventricular septum. Here's the left wall of the heart. This is the right wall of the heart. These are the papillary muscles. We have 2 on the left, 3 on the right. Why? Because the valves, they have different number of leaflets. These are the atrioventricular valves. The one in the right side is called tricuspid for 3 cusps. And then the one on the left is called bicuspid, or also known as mitral valve. It has 2 leaflets on the left, 3 leaflets on the right. And here are the chordae tendinae. So these are the flaps of this tricuspid and mitral, and these are the chordae tendinae. And these muscles in here are the trabecular carni. And here's the pulmonary trunk with this valve that has 3 leaflets. So this is the pulmonic or pulmonary valve. And here we have the aortic valve, which also has 3 leaflets or 3 cusps. So the tricuspid and the mitral has different number of valves, and these are called atrioventricular, and these are the ones that are called semilunar, because the leaflets, they look like half moon shape. Lunar means moon. And then out of these ventricles, the blood exits then through these semilunar valves. And this is a specimen showing you the same structures that we saw before. So, functions of these atrioventricular valves prevent backward flow. And in this case, the pressure in the ventricles when they contract is very hard, that pushes the blood back into the atria. And these leaflets close, so preventing this one-way flow. So tricuspid is located on the right side between the right atrium and the right ventricle. Bicuspid or mitral is in the left side between left atrium and left ventricle. Semilunar valves. Each of them normally has 3 leaflets. They look like half moon shape, and there is a reason for this. So, with this half moon shape, as the blood goes up, when the ventricles contract, and then this will, let's say this is like the lateral view of this or frontal view of this, as the blood goes up, flowing in this direction, by gravity, the blood will tend to go down and start sinking in here, and that closes the leaflets. So blood goes up, then by gravity goes down, and then you have weight in these valves, and then they close. And then, once the blood exits the ventricles, it doesn't come back to the ventricles. So it's always giving you one-way flow. Sometimes these valves, either atrioventricular or the semilunar, can be affected, like from calcification or from infections, and they need to be replaced. You can insert a mechanical valve. Now, in the case of the aortic valve, sometimes the person, instead of having 3 leaflets, for some reason you have 2, and in this case you call it stenotic aortic valve, and then also needs to be replaced. So, difference between the atrioventricular and the semilunars is that these semilunars are not attached to chordate tendinae and less to pectinate muscles, because they form the end point of exiting. So there is no need to contract to bring them back into the ventricles, because they never go into the ventricle. They just close, they hold that blood, and they keep it like there. So this is the superior view of these valves. So these 2 large valves are the tricuspid on the right side and bicuspid on the left. So this one has 3 cusps, and this one has 2. They're attached into this fibrous ring. And then here are the semilunar. The one that is closer to the AB will be the aortic, and the one that is most posterior is the pulmonary trunk. Or sorry, more anterior, pulmonary trunk. Each of them has 3 leaflets. And this is the actual view of one valve, like in this case is the pulmonic open, and this one is closed. So sometimes the valve fails to close. In this case, it's going to be called insufficient. So some blood will go into the atria. Sometimes can be stenotic, narrow, because of calcification or a genetic defect. And instead of having 3 semilunars, you have 2. And no matter what the defect it is, you can have an extra hard sound, which is going to be called a heart murmur. And sometimes you can have enlargement of the heart, especially in stenotic, because the blood has to pass through a narrower canal. Sometimes you can have fatigue, dizziness, and you can even faint in the case of stenotic valves, because you have less oxygenation, because less blood flows through that stenotic valve. And then you can have heart palpitations. The most commonly affected will be the mitral and aortic valves, the ones in the left side. So any questions? Now, here we have blood flow put it all together. So blood goes into the lungs to be oxygenated, and then once the blood is oxygenated and it goes in this direction from the right side into these lungs, it will return to the heart oxygenated into the left side so that it can go and deliver this blood through the systemic circulation into the capillaries of every tissue in our body. So here it is, the going back into the systemic circulation because you have contraction of your left ventricle and deliver of this oxygenated blood into the aorta. Now for the coronary circulation specifically, so the right and the left coronary arteries are the first branches of the aorta, the ascending aorta specifically, the little part that goes up. And the right coronary is longer than the left. Both of them, they run in the coronary sulcus, the little indentation between the atria and the ventricles externally along the surface of the heart. And then the right coronary artery is going to give different branches, including the one that supplies the right atrium and the ventricle with this branch that is called the marginal artery because it runs on one margin or one edge of the heart, and the posterior interventricular that runs in the interventricular septum, sulcus posteriorly in the heart, which is basically the equivalent of the anterior ventricular artery, which is branch of the left coronary artery. So here it is, left coronary artery has the anterior interventricular artery, which is also called left descending artery or LAD. And before it gives you, after it gives you this branch, it also gives you this circumflex, which is the one that goes around to go into the posterior part of the heart. One way to visualize these arteries is by an angiogram. An angiogram is basically injecting a contrast material through the veins, and once this contrast material reaches the heart, you can see how this blood goes into the heart. So in this case, we have the left coronary artery, and this will be the anterior interventricular artery, and this is the circumflex, and there is a lack of flow of this material in here. So anything that you see in white color represents the blood vessels. So there is an obstruction here in this left coronary artery, providing less blood supply to this. So this is called specifically coronary angiography. And then you can do these angiograms of other blood vessels, including the ones that provides blood supply to our brain. And then you can potentially predict then what can be the consequences. So once the blood in the heart goes to the different branches of these coronaries, the left and the right, they return into the coronary sinus by these cardiac veins. So we have the great cardiac vein that drains the left side of the heart, the left atrium, and much of both ventricles, but especially the left. And then we have the small cardiac vein that drains the right atrium and the right ventricle. And then we have the middle one that drains the posterior part of the left ventricle. So sometimes you have variation again between individuals. So you can have anastomosis, and then you can have collateral circulation as well. Anastomosis is when two blood vessels merge, like for instance between the coronary arteries or arteries outside of the coronary circulation. And in this case, you can have collateral circulation, helping to prevent only one branch to be in charge of providing blood supply. especially when the heart, it has an obstruction internally. That's when more than likely this collateral circulation can develop. So here is the coronary circulation then. So here we have right coronary artery. This is the marginal artery running here on the right side of the right ventricle. So this is the area that is supplied by this right coronary artery in this color. And then in this yellow color, we have this area supplied by the anterior interventricular artery. All of the left ventricle anteriorly and part inferiorly, while the circumflex supplies the left atrium and the posterior part of the left ventricle. And then here we have the posterior interventricular artery that is branch of the right coronary artery that provides blood supply to this posterior part and this anterior part of the right ventricle together with the right atrium. And then for the veins, so this is the color code for what each of these veins drain. So the great cardiac vein is called great cardiac vein because it carries most of this blood supply from the heart. So it runs together with this anterior interventricular artery in the anterior interventricular sulcus and basically drains the left atrium, the left anterior part of, the anterior part of the left ventricle and this margin of the left ventricle. The middle cardiac vein drains this middle part in between the edge of the left ventricle and part of the right ventricle. And then this part in purple color is drained by the small cardiac vein. The right posterior edge of the right ventricle and this right atrium. So this is the small cardiac vein in here. And all of them ultimately, no matter if it's the great, the middle, or the small cardiac vein, they drain into the coronary sinus that drains together with superior and inferior vena cava into the right atrium. So, problems with the coronary circulation. We can have CAD or coronary artery disease, which is basically a spectrum of medical conditions, including total or partial obstruction of these coronary arteries and its branches. If you have total obstruction, you will have no blood supply to a particular area of the heart, and then you will have necrosis or death of the muscle tissue, and this is called myocardial infarction or MI. If it's partial, you will have something that is called angina and angina pectoris. This can be basically very severe or less severe, and then the main symptom is pain in the precordial area, in the area of the heart, that sometimes can be aggravated with exercise, and sometimes the pain doesn't go even with rest. So depending on the problem is the treatment. Risk factors for CAD include smoking, high blood pressure, poorly controlled diabetes, and high levels of certain lipids, cholesterol specifically, obesity, age over 40 for males, and 50 years and over for females. Genetics as well runs for CAD in families, and ethnicities. In males, they have greater chances of having CAD because in females, at least before menopause, they have high levels of estrogens, which forms a protective factor for having CAD. So those are the risk factors for CAD. Treatment will be coronary angioplasty, in which they introduce a balloon through a catheter into the area of obstruction directed with an angiogram, and then they inflate that balloon, and they can break down these deposits of calcium and cholesterol and other lipids in these arteries. Or they can bypass. So if the obstruction cannot be relieved, they can take a piece of vein, one of these major veins, like the greater saphenous vein in our leg, and then they can put it in between the obstruction, well, in between the normal area and the obstructed area, so that the blood flow can go from normal area into the area that it was obstructed, so that person can have an alternate route of blood. So any questions about coronary circulation? Okay, now for physiology of the heart. So our heart beats automatically. It doesn't require input from the nervous system. We have specialized cells that are called the pacemaker cells that undergo spontaneous action potentials. So this property of the heart then of beating without the aid of the nervous system is called autorhythmicity. And this autorhythmicity will be controlled by the nervous system, so that we don't have basically more heartbeats than usual. So we'll see how this happens. Some of you might have seen documentaries or movies in which they're performing an open heart surgery and a heart transplant, and they take away the heart from the person and they put in a tray, and then the heart still is beating outside of the body, and then they put a new heart. Is that science fiction? No, it happens. As long as the heart has the right number of ions, right temperature, and it has right pH, the heart will keep beating, and it has oxygen and all that. Eventually the heart will stop beating, of course. But basically that's autorhythmicity, the property of beating on its own. Now, histology of the heart muscle. A heart muscle is different from a skeletal muscle. It has cylindrical shaped cells just as a skeletal muscle, but these cells are shorter and they branch. Skeletal muscles, the cells are longer and they don't branch. And in the heart muscle, we have one nucleus per cell. In a skeletal muscle, we have more than one nucleus. And we have striations on both that gives a specific pattern that you can see in the microscope, but the heart muscle has stronger striations that we call intercalated discs. And these intercalated discs are nothing else but strong connections that prevent the heart muscle cells from pulling apart when the heart is contracting. And also these muscle cells in the heart, they have this physiological property of contract basically without the command of the brain. So they are autonomous. So this is involuntary muscle. In a skeletal muscle, you contract it with your will. So this is voluntary muscle. Now, something that also is particular to this muscle, the cardiac muscle, is that it has abundant myoglobin because it requires a lot of oxygen. Which is the muscle that contracts 24/7, guys? Out of smooth, cardiac, or skeletal? The cardiac. So it requires more oxygen than any other muscle. And it has to have a lot of mitochondria to produce energy. And then it has also a lot of glycogen, so that if it runs out of glucose, it has its own supply of glycogen. Now, within these muscle cells, we also have gap junctions. If you remember from AMP1, gap junctions are little windows in between the cells that allow the transfer of ions, so that each of these cells on the sides, they have equal number of these ions. So one property of the heart is that it contracts in unison, contract all at once, so that they can have a strong force and that they can propel the blood into the areas without a problem. So they are what we call a functional syncytium. And within the sarcolemma or the plasma membrane, remember sarcolemma is the proper terminology for the plasma membrane of muscle cells. So within the sarcolemma, we have gated ion channels, like sodium channels, that basically open when you have a specific voltage. And then we have calcium channels that opens when you have a specific voltage, and they close after a certain period of time, like within 100 milliseconds, for instance, or 150 milliseconds. And then we have potassium channels that are either ligand-gated or voltage-gated. Voltage-gated, again, is when you open at a specific volts, at a specific electrical voltage. And then ligand-gated is when they're bound by a neurotransmitter, for instance. And then we have non-selective cation channels, or HCN, that basically can be activated when you have hyperpolarization. If you remember from action potentials, we have several phases, including depolarization. So that means when the cells are less negative, repolarization when the cells return to negative, and then hyperpolarization when they become more negative than usual. And that basically helps for these sodium ions to enter or the potassium ions to exit simultaneously, these channels, the non-selective. Why they are called non-selective? Because in this case, both sodium and potassium can come in and out. So here's a view of these muscle cells. So this strong pattern in here, bands, is the intercalated disc. This is the nucleus, and you cannot see really well the striations that are less stronger than the intercalated disc, but it's where the myosin and actin bands extrapolate, or maybe not, depending on what area you're considering. And then if you magnify and make a cartoon, basically this is the intercalated disc. This is a branching of these cells, and if you see inside, you will see abundant mitochondria, and then you can see the gap junctions helping to transfer these molecules, these ions, so that the heart can contract at once. So, reviewing electrophysiology. So voltage, what it is the difference in the electrical potential between two sides or 2 points. Membrane potential is the voltage that you have when the cells are at some point either at rest, and you call it resting membrane potential, and then you have reversal of this potential, which is typically minus 60 to minus 90, depending on the cell. And then you have reversal when you have an action potential, and then you have return to the normal membrane potential that you have when the cells are at rest. And then you have the ion gradients, basically the separation of ions across this membrane that allows this flow of ions in and out of the cells. That is what keeps these cells to be excitable. Typically, we have classes in this classroom during the weekdays. You guys are the ions. You are the things that makes this classroom alive. You come in and out at a specific period of time during the weekdays. So we have an action potential during the weekdays. During the weekends, when there is no students, this room is so quiet. This will be the resting member potential. There is no activity because there is no ions. Okay, there is no exchange. So, current is the flow of ions or electrons with a chemical or an electrical gradient. Less students in the hallway, more students in the classroom, inside the classroom. That's a gradient. And then depolarization, the change in the resting member potential from minus 60 to minus 50, etc. So less negative. That leads to a threshold. To reach that threshold, and then you have an action potential. For some reason, it was a weekday, we have a scheduled class, but only 2 of you show. There's no enough ions. Should we have class? Maybe not. We canceled class in that case, so there is no depolarization. And then we had the class normally. It's over, it's the time for go, you won't stay longer than that. Then you exit, then this classroom become, again, negative, so this is repolarization. Okay. So that's usually when potassium ions leave the cell. Potassium ions are positively charged. Hyperpolarization, when you have less than usual, when you have more negative voltage in your cells. And this can be because your potassium channels remain open longer than usual and you have more exiting of this potassium. Can that return to normal? Yes. At the beginning of the pandemic, we had classrooms that they had less number of students than you guys are now. But as time passes by, we have more number of students. So that's returning to normal. What maintains this gradient is different than sodium-potassium pump, which brings out potassium in exchange for sodium. And that is maintained by ATP, adenosine triphosphate. And then also we have a transport pump for calcium. So any questions about the review of electrophysiology? Okay. Now, the heart has this specialized system that maintains auto-rhythmicity that is called the conduction system. And it consists of several parts, groups of cells that are not muscle cells. They are specialized cells that are capable of generating automatically these action potentials. So we have a group of cells that are located in the nearby part of the superior vena cava. This is the SA node. And this is the pacemaker specifically of the cell. This pacemaker has a membrane potential, a resting membrane potential from minus 60 that can go and revert up to plus 10 millivolts automatically without the input of the nervous system. As long as you have concentration gradients of calcium, sodium, and potassium ions, you will have this exchange of ions across this membrane of these cells, and then you have generation of an action potential. Mainly will be because you have leaky channels for potassium. And then we have the pacemaker potential that occurs slowly because you have lack of voltage-gated sodium channels within these cells, but you have, again, these leaky channels that are non-specific, but they respond more to potassium than sodium. And then you have slow depolarization, as opposed to rapid depolarization in skeletal muscle, and also in cardiac muscle cells. So what are the parts of this action potential in this pacemaker cells. So you have a slow initial depolarization that begins in this minus 60 millivolts, and then once you reach minus 50 millivolts, the threshold, then you have a full depolarization, which actually, the threshold is minus 40. So you have full depolarization phase. When you have full depolarization, you will open your voltage gated calcium channels, and then calcium will enter the cell. And that is going to open in your cardiac muscle cells voltage-dependent calcium channels that will trigger a stronger contraction of the muscle cells in the heart. So these are the phases, and these are the channels that participate to make these phases to happen. So this represents the plasma membrane, and these are the channels in the plasma membrane. So we have the non-specific channel that allows sodium and potassium to come in. It's basically a cation channel. And then here we have the calcium channel and the potassium. So here you have your resting membrane potential minus 60. And then slowly through these inspecific or non-specific channels to be open, leaky channels, you have channels coming in and out, more positive coming in than out. That leads to accumulation of positive ions and slow depolarization from minus 60 to minus 40, reaching the threshold. Remember, in electrical physiology, once you reach the threshold, you are in the all or none rule. Basically, you create an action potential no matter what, okay? So that will open then your calcium channels once you reach the threshold, and calcium, extracellular calcium, will come in, and that will lead to the full depolarization phase. So from minus 42 plus 10 millivolts. So in this graph, what you can see in the x-axis, in the y-axis is the millivolts from minus 60 to plus 10 millivolts, and in the y-axis, you have the time in milliseconds. So you start in 0 milliseconds, and when you are in 400 milliseconds, you have reached your full depolarization. So your cell from being negative becomes positive. And then you start the repolarization phase, which basically happens 100 to 150 milliseconds after the calcium channels have opened. And then you close these calcium channels, and then you open your potassium channels. So potassium will exit, and then no more calcium enters. So your cells from plus 10 gradually become more negative because you lose your positive charges. And then you have a minimum potential phase in which the potassium channels remain open until you reach this minus 50. And then if these potassium channels remain open longer, from minus 60, you can go up to minus 70 or up to minus 80, and that is called hyperpolarization. And that's when your inspecific or non-specific channels start to open, and you start the cycle again. So it's a cycle. So this is the repolarization phase. Your calcium channels close, your HCN channels are also closed, so potassium exits, and you lose positive charges from plus 10, you return to minus 40. If this one remains open, the minimum potential phase, you go from minus 40 to minus 60, sometimes up to minus 70, minus 80. And then you close this one open, they become activated due to this hyperpolarization, and then you start the cycle again. And then you keep going like that. Slow depolarization, full depolarization, repolarization, hyperpolarization, and start the cycle again. So this is what we call the pacemaker action potential, which is different from the other two action potentials, the cardiac muscle action potential and skeletal action muscle potential. So now for the segments. So we have these segments SA node that is located in the right atrium, lateral to the opening to the superior vena cava. This one depolarizes or creates action potentials at a rate of 60 to 70 or more times per minute than the normal cells. So this is influenced by the sympathetic and parasympathetic nervous system. SA node basically, guys, depolarizes so fast that if we didn't have the action of the vagus nerve, which is part of the parasympathetic cranial nerve number 10, we will have a heart rate of 100 beats per minute. And that's tachycardia. So because the vagus nerve helps to drop this heart rate to 70, this depolarization to 70 beats per minute, or 80 on average, basically, we have less beats per minute than tachycardia. And that is good for us because that allows the atria to get filled with blood and to transfer normal amounts of blood to the ventricles. Every time that someone has tachycardia, the heart has less time to feel its purpose, which is pumping a specific amount of blood per minute. And that reduces the life of a person. It is well known that bradycardic species, a species that has a lower heart rate than others, they live longer than a species which has higher heart rate. Like, for instance, mice, they live longer than humans. And then turtles, they live longer than humans. Why? Because they have lower heart rate. And it is well known that among humans, if you are a stressed person, if you have tachycardia a lot, you're going to live less longer. Because we have a finite number of heartbeats per minute. And now, the SA node sends the signal to the side so that the atria can contract through the internal pathways. And then the SA node, after that, sends the signal into the AV node, which is located at the base of the tricuspid valve, posteriorly. And this AV node basically makes our heart to beat 40 to 50 times per minute. So it has a slower heart rate than the SA node. So when someone has a problem with the SA node, the regular pacemaker, the next one that take over the role for making the heart to contract will be the AV node, and that is not enough to send blood supply to the brain, and then the person starts feeling dizzy and ultimately will faint. And then the person has to have a pacemaker so that you can increase the heartbeat so that the blood supply goes into the brain, which is very important to keep us conscious. So that's the AV node. Now the next is going to be part of the conduction system, is going to be the Purkinje. But before the Purkinje, we have the atrioventricular bundle that separates into the right and left bundle branches, and these run in the interventricular septum. The right bundle branch is going to provide this conduction system to the right ventricle, while the left to the left side. And then from there you have the Purkinje fibers that goes up from the apex into the base of the ventricles so that you have kind of a wiring system that carries these action potentials so that every single cell, muscular cell in your heart contracts. I compare this conducting system to wires in an electronic device, or let's say even your car. If you damage these wires to a specific part of your heart, of your car, that part of the car won't work because it's not receiving these electrical signals from the battery. It doesn't run into your lights, into your honk, anything. So similarly, if our conducting system will be damaged, you will have consequences. These are called blocks typically. So you can have right bundle, left bundle, branch block, etc. So when your SA node is functioning and sending electrical signals so that the rest of the conducting system makes the heart muscle cells to contract, you will have a beat or heartbeats in your heart that will be reflected in an electrocardiogram, and this will be called sinus rhythm. It is shown by the presence of a wave in electrocardiogram. We will see that later. It's called the P wave. So every time that you see a P wave in an electrocardiogram, you will be certain that the SA node is making that electrical signal. Okay? And then SA node will transfer that electrical signal to the AV node, to the bundle branches, and then to Purkinje fibers. And sometimes when you have a block in any of these, any other pacemaker can take the lead. And this is called ectopic pacemaker, but that will change the rhythm of the heart. It's not only having the normal number of heartbeats, but the time interval between the heartbeats is also important. It's like most of us have 3 meals per day at a particular period of time. It is kind of not good for the body to, well, skip meals and let's say, well, I didn't have any meal today, tomorrow I will have 6, and at the same time. No, you have to have periodicity so that you can absorb the nutrients. So same for the heart. We have to have regular rhythm. So SA node generates an action potential which spreads to the atrial cells and the AV node and requires 0.03 of a second. And then after, you will have the action potential reaching the AV node, and then you have a delay. That delay will allow the atria to get full with blood and then contract and pass the blood into the ventricles. And that can also be seen in the electrocardiogram, that delay. We will see it later. So that delay lasts up to 0.13 of a second. And then your Purkinje fibers will transfer then the action potentials after you receive it from the bundle branches right or right and left, into the rest of the cells in the ventricles. And this speed, which is 150 times faster than the cells of the AV node. So once your ventricles have the blood in it, they push it out faster because of this speed that the Purkinje fibers transmit the action potentials. So these leave a number of gap junctions in their membranes that is larger. That's why they can transfer the action potential much faster. So these last between 0.6 of a second up to 0.06, sorry. So that total action potential duration in the heart is 0.22 seconds. So here are the parts of this conducting system. AV node internodal pathways that transfer the action potentials. You can see the arrows into the right and left ventricle muscle. And then here we have the AV node. And then here we have this AV bundle that subdivides into the left and right branches. And then at the apex, you have the Purkinje fiber that transfer the action potential fast into the base of the ventricles. Any questions? Okay, so this is all for today.